Dear students, in this module we'll be talking about genetic code, which is defined as the relationship between the sequence of amino acids in a polypeptide and nucleotide sequence of DNA and mRNA. DNA is the director which controls and regulates and determines the nature of protein that needs to be synthesized in a cell. Talking about the genetic code, the process of translation requires transfer of genetic material from a polymer of nucleotide to a polymer of amino acids. The changes in the nucleic acids are responsible for the change in the amino acids in the protein which is to be translated. This led to the proposition of the genetic code that could direct the sequence of amino acid during the synthesis of protein. So the group of nucleotides that specify one amino acid, that is a codon, and the relationship between the amino acids in a polypeptide is what is referred to as the genetic code. So the interrelationship between the nitrogenous bases. There are 4 nitrogenous bases, A, T, G, and C, and the proteins are synthesized from different 20 amino acids, out of which some are essential and others non-essential. A singlet codon like A, T, G, C. Each base, it will specify one amino acid. So only 4 out of the 20. If we presume the genetic code to be doublet, we'll have just 16 of it. If we take it to be a triplet, we'll have 64. That means then the code will become repetitive and it suffices for 20 amino acids. And thus, the experimentation was carried out by Nirenberg, Mithai, Hargob and Singh Khurana to determine that the codon comprises of the triplets of the nitrogenous bases. So the triplets of bases, they make up the 20 amino acids. Type of codon, number of bases, and let us take the number of codons. So if the code is singlet, the number of bases 1, codons will be 4. Doublet, number of bases 2, codon 16. If it is triplet, it will be 3, number of codons 64. That suffices the purpose. So in this module, we had a discussion on genetic code. Thank you.